All right, so this is the second half of respiration, so kind of in and out. Uh, Next up in your notes, you see is the neuronal control of breathing. I want to go through a couple things, but again, the most important thing you have to really note is what's driving you to respire. People always think it's a lack of oxygen. No, because again, your blood doesn't want oxygen, it wants to release it. Kind of like I said, like a soda releasing gas whenever it goes flat. Um, the major thing that drives you to breathe is actually an excess amount of carbon dioxide in your blood. Why? Because the thing that I drew for you before, the reaction scheme, was carbon dioxide will combine with water to make carbonic acid. That acidity is going to be picked up by your brainstem, specifically the medulla oblongata, and therefore that's going to actually force you to breathe in. So people always think that it's a lack of oxygen. No. It's going to be an excessive amount of carbon dioxide. That's which causes carbonic acid. That's what's going to first make you want to breathe in. Secondly, as a secondary factor, obviously, is if you have excessive CO2, that would also create higher acidity levels. So the second factor that makes you breathe in is a drop in your blood pH. Why is this so effective? Because in your brainstem you have cerebrospinal fluid which surrounds it, and in that cerebrospinal fluid you don't really have any buffers to pH. So any small, tiny change in pH is picked up in a second, and therefore your brain responds hopefully accordingly. Why do I keep focusing on the brainstem? Because the brainstem evolutionarily is the most primitive part of your brain. It basically covers your complete base life abilities, meaning your blood pressure, your heart rate, your respiratory rate, the things that keep you alive. That's the brainstem. When people say like, oh, somebody's in a coma, are they ever going to come out, or they're brain dead, will they ever come out? Depends upon where the damage is and how excessive it is. However, if the brainstem is damaged, the person's never going to be able to respire or control their heartbeat on their own, so therefore they would have to be on a machine for the rest of their existence. And that's, again, the person's choice whether or not to keep them on or, quote, pull the plug. All right, so neuronal controls of breathing, as you notice, understand number one is controlled by levels of carbon dioxide, specifically excessive carbon dioxide, which would then, number two, create a drop in pH. That's the major take home I want you to come with this. You have in there then, understand, I'd expect you at this point in your life and education to know what hyperventilation is, but I need you to understand it on a biochemical level. Think, when you're hyperventilating, you're going, what are you actually doing? You're expelling carbon dioxide at a faster rate than your body can make it. Well, if you're expelling CO2 at a faster rate, that means you can't make carbonic acid in your blood. So if it's not going to be an acid in your blood then becomes more alkaline or more basic. So it becomes a higher pH, higher than 7.45. When that happens, blood vessels leading to your brain will constrict. When they constrict, that means then you don't have blood flow to your brain, and what's the major thing happens when you hyperventilate? You pass out. Right, that's your body basically hitting the do-over button going, okay, you can't control it, let's hit the start button, start this thing all over again, and let's see if we can handle this. But that's a hyperventilation. Think what the cure of hyperventilation is. You breathe in your own carbon dioxide. Right, because you're giving it off too much, you're putting it back in to build up acidity so the blood pH comes back down to homeostatic parameters. All right. So again, all unconscious breathing is going to occur with the medulla oblongata. You see in the notes that there's three major areas in the medulla called the dorsal respiratory group, the ventral respiratory group, and the pneumotaxic center. Thing you really have to focus on is this. Inspiration, breathing in, is an active process. What I mean by that, there has to be a brain stimulation, there has to be an action potential, a nerve impulse, which causes the diaphragm and the intercostal muscles to actually contract. So inspiration is actually an active process, so it needs ATP, it needs stimulation. Expiration, breathing out, is just stopping the signal. So really it's more of a passive process where everything just relaxes, goes back. And so in your brain, what you have are inspiratory neurons because it has to be activated. As far as expiratory neurons making you breathe out, they're there, but really only when you do forced expiration, when you go and actually push out. It's not really, necessary, quite frankly. So, the dorsal respiratory group, again, we're talking about the brainstem, back of your head. The dorsal, which means facing the back, that is called those who have the inspiratory neurons. Now the ventral, which would be facing, the back of your head, back here. Therefore, the ventral one has both inspiratory and expiratory neurons. Why? That's whenever you have heavy breathing, like forced respiration, like I was saying. The pneumotaxic center, found within the brainstem, actually coordinates the change from inspiration to expiration. So breathing in, breathing out. It's a coordination group. All right, so as I have in there, again, carbon dioxide, hands down, is the main reason why you breathe in. Again, excessive amount. Secondary would be a drop in pH, more acidity. Drop in pH with more acidity because of carbonic acid. Oxygen, however, minimal to any change in respiratory rate. However, there are cases where somebody has a chronic, obstructive pulmonary disease, a COPD, where it's called a hypoxic effect, where the actual lower drop of oxygen will actually force them to breathe. So, but that's not the normal. That's one of the outlier cases. Okay, so popping on down there, big huge star where it says chemical and peripheral input to respiratory centers. There's central chemoreceptors, peripheral, Herring-Brewer reflex, and irritant receptors. Know all four. Guarantee you'll see them in one form or another. The central chemoreceptors are specifically, they're called central because they're in your central nervous system. They're in the brainstem. Those are the ones which are primarily going to react due to a drop in pH, due to, again, excessive CO2. Again, because there's very little buffers, it picks it up in a heartbeat. Peripheral chemoreceptors means protein receptors which are found outside of the central nervous system, which can also help regulate breathing. Big star, where do you normally find them? You find them in the aortic and the carotid bodies, meaning in the aortic artery, the carotid artery. Those ones, big star, they do actually respond to differences in oxygen levels. One of the few that would actually do that. Herring-Brewer reflex is a overstretch reflex. Think if you ever breathe in, you breathe in as high as you can, you go, then you can go a little further, and then eventually you have to stop. Your body won't go further. That is called a stretch reflex. That's the Herring-Brewer reflex, which prevents overstretching of your lung material so that you don't rip it. So that one's stretch receptors. Irritant receptors you have everywhere where you have the pseudostratified ciliated columnar epithelium. That's why you sneeze when you come in contact with dust, why you cough when there's smoke. Those are irritant receptors forcing out if you get large particulate matter and try to push it back out either through coughing or sneezing. And again, at this point in your education, I would expect you to know between a cough and a sneeze. You're like, I know what that is. Think what it is physiologically. When you cough, first thing you do is you go, huh? What did you just do? You closed your glottis and then you forced air out. When you sneeze, you go, chew. The glottis is wide open now. So the difference between a cough and a sneeze is the glottis open or closed before you force air through the larynx. Now the ventral part of the medullary rhythmicity area, the dorsal part, understand, is for unconscious breathing. The ventral part of the medullary rhythmicity area in the brainstem is basically so you can hold your breath. You all know that you can go and hold your breath, but you can hold it for only so long. Why? Because the subconscious part of your brainstem goes, oh, well, you're being stupid, and it just forces you to breathe again. Well, how do you hold your breath? You have to bypass your brainstem. You have to use your somatosensory cortex. You run the nerve impulse down your face with phrenic nerve number 8, which then takes care of the diaphragm, preventing it. The perfect example of that is a hiccup. When you get a hiccup, what you're doing is that phrenic nerve has been agitated. It misfires, so you go, Think of the cures of hiccups. You hold your breath. You drink water. You become frightened. You do some physical activity or flip upside down, do a handstand. Understand, think what all those things are doing. When you hold your breath, you're not expelling CO2. It's building up. Acidity drops. When you're drinking a glass of water, you're not drinking. Acidity builds up. All of them involve increasing the blood pH, decreasing the blood pH, increasing the acidity. What that does is it deadens the nerve and your hiccups go away. All right. So, coming up next then, we come to, I can't stress this enough, one of the most important things you're going to see again are going to be the gas law. So you see there Boyle's, Charles, Dalton's, and Henry's law. Boyle's law and Dalton's law I talked about in the previous lecture, but let's go over it right now. Boyle's law says that the pressure of a given volume of gas is inversely proportional to the volume of the area it occupies. So they're inversely proportional, meaning pressure is going to be inversely proportional to volume. Okay? So what that means then is we have an inverse proportion, and therefore meaning that as volume decreases, pressure increases. As volume increases, pressure decreases. One goes up, the other goes down. That's when I was saying why you don't puncture the can, the aerosol can, because if you puncture it, the volume decreases, pressure goes up, and kaboom, it explodes. All right, Dalton's law says that temperature and the pressure of a gas are going to be directly proportional. The temperature and the volume of a gas are directly proportional. So now temperature is going to be directly proportional to volume. Meaning, as temperature goes up, volume goes up because you know it expands. Temperature goes down, it contracts, volume decreases. Now, Dalton's law. Dalton's law is called the law of partial pressures. This becomes more important when we talk about the gas exchange in the alveoli. What Dalton's law of partial pressure says is that if I have a mixture of gases, like you're breathing right now, nitrogen gas, oxygen, water vapor, plus trace gases, inert gases, xenon, hopefully not radon, but let's see, carbon dioxide, all that, it's all around you right now. But what Dalton's law of partial pressure says is that the total pressure exhibited by a mixture of gases is equal by the pressure of the first gas plus the pressure of the second gas plus the third, and so on. Meaning that what you would do is you would add up all the individual pressures of the individual gases. When you add them up, that would be the total pressure exhibited by the mixture. Okay, fine. But, very important, each one of these gases is going to behave chemically, physically, like none of the other gases are present. What do I mean by that is go back to when we talked about diffusion. It's always going to be high to low, meaning high pressure of a gas to low pressure of a gas, irrelevant to the other pressures of the gases around it. It acts independently of the others. That's for the gas exchange in the alveoli. The last one, Henry's law, says this. Now, it comes out very technical, but it becomes very simple once we break it down. Henry's law says this. The solubility of a gas in a liquid is dependent upon the solubility of that gas in the liquid and the partial pressure of that gas exhibited over the liquid. You're like, that's a lot. Well, think. I'm assuming you purchased a 2-liter bottle of soda pop before, right? Okay. You understand to make the soda pop bubbly, what they do is they take the sugary solution and they inject carbon dioxide into it. Now, when it's going to be shipped out and bottled, if you ever notice, your 2-liter bottle, your brand new bottle, has that little space above it. And people always say, oh, well, they're cheating me because, I'm not getting the full amount. You're getting every little micro pen. No. Think. The carbon dioxide doesn't want to be in the mixture. It wants to get out. So what do you want to do is you want to build a cap of pressure of the same gas over the liquid to hold it in. Think, the first time you open up a 2-liter, you hear, pshh, that's all the extra carbon dioxide that was pumped in on top of it, keeping your beverage carbonated while it was sitting on the shelves for how many weeks, months it was there. But the second you remove that cap of pressure, think, it starts to then lose more gas, and the more you open it, the less time you hear psh, psh, less and less, and eventually it goes flat. That's why if you keep soda in a refrigerator, that's why it stays longer. The refrigerator gives the gas less kinetic energy, so it doesn't have a chance to get out as much. But as you know, if you manually shake it, well, that makes the gas come out faster. Right. That's all Henry's law. Applications of Henry's law. Very large star, very important. Decompression sickness, meaning the bends, and nitrogen narcosis. The bends. So, if you know anybody who scuba dives, if you've scuba dived yourself, keep in mind, as you go down in the water, for every 10 meters you go down, your pressure increases another atmosphere. Now, currently right now, what you're breathing in is a mixture of gases, primarily nitrogen gas and oxygen gas. Those are the two major players. Your body doesn't want nitrogen. So as you breathe it in, roughly you're looking at, your amount of the percentage of nitrogen in the atmosphere right now is in the 70s. Oxygen is around 21%. But the amount of nitrogen coming into you and coming out of you is roughly the same, meaning your body's like, it comes in, I don't want it, pushes it back out. Well, you understand oxygen, when it comes in, very little comes back out. That was the point of hemoglobin from the previous lecture. All right, it basically has like a sponge to attach it and move it on. Now, Problem is this: when you go down, 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 down in depth, and let's just say in your tank you have a regular air mix, just like regular atmosphere, your body doesn't want nitrogen. But think, as you go down, 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 down in water, you're going deeper and deeper into a bigger soda bottle. The cap above you in pressure keeps becoming greater and greater. Nitrogen doesn't want to be in you, but the pressure around you is going, you're going in and staying in. So as you go down deeper, say going over 150 feet, 180 feet, you're taught you don't stay down there very long. Why? One major factor is you get excessive nitrogen in your bloodstream and you get something called nitrogen narcosis. So let's just say you're 130 feet down underwater. Nitrogen narcosis gives you an intoxicating effect, like you're drunk. You really don't want to have that feeling when you're 130 feet below the water, because then you won't have your orientations, you won't know what is up, what is down, and you're going to die. Now, when you're done, do you pop up like a cork? No. When you're down for a certain time, you have safety stops. You have to come up, stay for a little while, go up a little higher, go for a little while. What you're doing is it took time to get the nitrogen into your bloodstream. It takes time for you to get it out to your lungs and breathe it back out again. If you pop up too quickly, the nitrogen gas in your blood is going to expand very rapidly. Think about taking like a small ball, like a dog ball that's not solid, but it's air in the middle. If you took it to the bottom of a pool, you notice that ball shrinks. But if you let the ball go, when it hits the surface, it expands again. That's what would happen in your lungs. But the problem is that the nitrogen gas expands too quickly in your bloodstream, it'll cause bubbles. Those bubbles will then cause what is called decompression sickness, also known as the bends. You get massive amount of pains in your joints because the bubbles get caught in capillaries, and basically you feel like you're in the fetal position in massive pain. It'll become terminal if it creates an embolism and it goes to your brain or your heart, and it'll kill you. How do you prevent that? Safety stops or a decompression chamber. So one question people always ask is like, can I get the bends or decompression sickness on dry land? Yeah. How? Let's just say you're a coal miner and you work in a vertical shaft and you go down a mile. Well, understand, as you go down, you're getting all that pressure built above you. It's just the same thing as if you were scuba diving. So if you ever follow anybody, that was saying coal mining or anything like that, when the cart goes down, it goes down pretty darn quickly. And when they're down there working, they can only allow them to work for a certain amount of time. Again, nitrogen narcosis, also things like, coal dust and whatever. But in the same token then, when they go back in the cart to come back to the surface, it's a much slower ascent because, again, you want to get the bubbles out. All right. So that's my talk on the gas laws. Know the gas laws, be able to compare and contrast them, and understand the examples I gave you. Remember, Boyle's law is the major reason why you breathe in. Charles's law helps to expand your lungs, but it's not as important as Boyle's law. Dalton's law of partial pressures is going to explain how we get gas exchange from the alveoli into the capillaries around it and vice versa. And Henry's law, easy way to remember it, think about that's the reason why soda pop goes flat if you let it sit out too long. That's basically Henry's law. All right, and the examples I gave you were decompression sickness and nitrogen narcosis. So let's see, let's skip down here then. We talked about inspiration already. I talked about expiration in the previous lecture. Pneumothorax means you get air in the pleural cavity between the lung and the rib. You didn't puncture the lung, but what you did is destroyed the pressure gradient, so the lung will collapse. Atelectasis is when the lung collapses because there's a blockage in the airway. Both cases, the lung collapses, but for different reasons. All right, let's jump down then. Next thing you see, resistance to airflow. Keep in mind, I said the bronchioles are very small, which is why when you have an inhaler, you use epinephrine, so if the muscle contracts, it can relax. Pulmonary compliance is a fancy-smancy word to mean how stiff is the lung, because the lung is supposed to be spongy. Well, if you have damage to the lung, let's just say you work in an asbestos factory, you smoke a pack of Luckies for the last 40 years, you work in a coal mine, you're going to lose elasticity of the tissue. So the question is, how elastic is it? How stiff is the lung? Also, other things you see in there that can cause a decrease in it would be black lung disease, from a coal mine or constant irritation, and tuberculosis. Tuberculosis occurs from the bacterium, Mycobacterium tuberculi. It's highly infectious and highly contagious. What does it do? It lives in the mucus of the lungs and basically will deteriorate the alveoli of the lungs as time goes on. So you get a persistent cough and a constant, what do you call productive cough, called sputum. It's the material that comes out. If it's not checked, keep in mind that it will start to cause massive deterioration of the alveoli. When somebody gets to terminal phase of tuberculosis, they have massive coughing fits, violent coughing fits, where they start coughing up blood in chunks. Well, what are the chunks? Those are chunks of their own lung that they're bringing up. So, that would be terminal at that point. So, let's see. Coming on down then, for number 3, for resist the airflow, I have the surface tension in the alveoli. Again, I can't stress this enough. Big star. The inside surface of the alveoli is supposed to produce a pulmonary surfactant. That's what makes the water more fluid. So therefore, when the air sac collapses, the water from one side of the air sac and the other don't touch each other, and then because of the polarity, keep the air sac closed. It makes the water more fluid, so if they come close, the air sac will reinflate. That's another reason for resist the airflow. All right, now we come down to ventilation measurements. Again, I can't stress this enough. Big stars. You've got to know these measurements. This is if you went to a pulmonary therapist. They would basically put a spirometer in your mouth, which basically monitors how hard can you suck in, how hard can you blow out, what is your normal rate, basically trying to assess health of the lung. All right, so the respiratory volumes, know them, memorize them, you'll see them again. So, first one you see there is TV. Know it by the abbreviation, know it by the actual name itself, tidal volume. Tidal volume means what you're doing right now. If you have a normal, healthy lung and you are currently inhaling, exhaling, inspiration, expiration, when you're not stressed, how much is coming in and out of you? A, quote, normal lung, you don't have to know the numbers, but the normal lung is roughly half a liter, 500 milliliters of air is coming in and out of you right now. Now, IRV is the inspiratory reserve volume. It's saying when you breathe in, when you go, how much more air can you suck in excess of tidal volume? So it's basically sucking in as hard as you can. Well, then expiratory reserve volume is when you go, how much can you blow out, forced out, then tidal volume. Okay? So, inspiratory reserve volume, again, don't know the numbers, for a normal healthy lung is between 2.1 and 3 liters. Okay? Now, expiratory reserve, notice how much you can blow out is only 0.8 to 1.2 liters. There's a disparity there. If you can suck in, say, 3 liters, but you can only force out 1.2, where's the rest of it? That's the important one. Next one down is called the residual volume. The residual volume is the amount of air left in the lung after your hardest blowout. Why? It's there so your lung doesn't collapse, and therefore it's always there. You always will have it unless you die. Think, you go to your stupid little soap opera shows or your doctor shows. The person is dying. I love you, baby. Is the residual volume. That's the lung losing the air and it's collapsed. Okay? But you only do that when you're dead. Now, all those, understand, are proportional to one's size, their body mass. In other words, if you're larger, you have larger volumes, smaller volumes. They're also proportional to male and female. Males tend to have higher volumes than females. Now, know those 4. Now, that's if you were at the doctor. Those are 4 pieces of data you would get from a spirometer. Now, the next one where it says respiratory capacities, you have to know the first 2. Vital capacity and total lung capacity. What these are calculations using the data from the first 4 measurements. Okay, so if you look, vital capacity is the amount that you can breathe in and breathe out with maximum stress. In other words, your deepest breath in and your hardest breath out. So what would it be? The inspiratory reserve volume plus the expiratory reserve volume plus the tidal volume. Those 3. Okay? Now, the last one, where you have total lung capacity, means I'm going to take your vital capacity, which is the other 3 numbers, and then I add it to the amount of the residual volume that you have, that dead space that you're not going to, that is your total lung capacity, okay? People always mess those 2 up. Vital capacity, always remember, is just basically your deepest breath in, hardest blow out. Your total lung capacity means that plus how much air is still left in the lungs that don't collapse while you're still alive, okay? So, coming on down, we have to understand there are what are called restrictive disorders and obstructive disorders. Restrictive disorders are things that reduce pulmonary compliance, like I mentioned, tuberculosis, black lung disease, say lung cancer from like asbestos, things like that. So therefore, they reduce they increase the stiffness of the lung, so it's not as spongy. Now, an obstructive disorder, this is now where we interfere with the airflow. This is where, big, huge star, you got to know what COPD means. Chronic obstructive pulmonary disease. I mean, the commercials are on probably 4 to 5 times a day. The old guy that can't blow out the candles and the kids going, well, Grandpa, you can't blow out. And they're doing the 3 little pigs. That's chronic obstructive pulmonary disease. That's going to be things like emphysema, chronic bronchitis, and brain fart. I'm forgetting the other one. Oh, Lord. Asthma. Asthma is the other one. All right. So, now, variations to respiratory rate rhythm. All those are different vocabulary words that, say, a pulmonary biologist would tell you to describe your rate and depth of breathing. I'm not going to go through all of them because they're just definitions. You can read them as well as I can. Quite frankly, if you understand, if I'm skipping it, again, put 2 and 2 together on an exam type of thing. I mean, it's not really that important. All right, so for the exam. Then I said, know what a cough and a sneeze is. All right, so air composition. You don't have to know the percentages of the composition of air. What I want you to note are the differences in the percentages. Atmospheric nitrogen is right around 79%, 78.6. In your alveoli, 74.9, meaning that your body doesn't want nitrogen. It's in, it's out. You look at oxygen. In the air around you, it's around 21%, but in the alveoli, it's 13.7. It dropped. Why? You're absorbing it into your bloodstream with the hemoglobin. When you look at CO2 and H2O, both of them are very low in the atmosphere, but when you look at them, they're both high, significantly higher in the alveoli. Right, because that's what you're breathing out. When you breathe out, you're breathing out carbon dioxide and water vapor. All right, so moving on. Let's see. You can read that as well as I can. I'm looking at the efficiency of gas exchange. Basically, how efficient you do the gas exchange depends upon gas pressure gradients outside your body and inside your body. One major thing you see there is altitude sickness. Again, understand, let's just say you decide to move to Denver, the mile-high city a mile up. For the first 2 months, 3 months, you're going to be miserable. Why? You're right now living at sea level. You're in Florida. So when you go, you have the entire atmosphere above you shoving air into your lungs saying, take it. When you're a mile high up, you have that much air, that much volume, and that much massive air pushing down on you, so then you don't get as much in your lungs. So you're going to have labored breathing. You're going to get headaches. They always tell you, drink tons and tons of water and take it easy. Build up. Why? Because as you're there doing normal activities at a higher altitude, your red blood cell content in your blood will start to increase. Erythrocyte populations increase. So even though you're getting less oxygen, there's more red blood cells, so therefore any oxygen you do get in, you can grab onto, and therefore you can start doing more physical activities. I always go to football. When the Denver Broncos always have a home game and you have somebody, say, from Oakland or Pittsburgh or Miami come in, you always see the game, this evenly matched type of thing. First quarter, third quarter, then all of a sudden fourth quarter, the visiting team is like sucking wind, right? Because their bodies weren't prepared for that, so they're on oxygen continually. Famously or infamously, there was a defensive back for the Steelers, and if I remember right, I'm trying to remember. I think he was, I forget what he had. I think he was heterozygous for sickle cell or something like that. Whenever they went to Denver, he could never play because if he would play, he'd die. So therefore, he couldn't because of the lower levels of oxygen. Whenever you see people like training for the Olympics or marathons or big races, what they'll do is they'll train at higher altitudes to build up their red blood cell content. So when they do the official race, they now have a competitive advantage over the others who didn't do that, and therefore they won't become as winded or as tired. With that then, we have gas solubilities, as I have there. Nitrogen, your body doesn't want. Oxygen, as I said, big star. Your blood does not want it. Your plasma doesn't want it. Think of it like a soda can. That's why big star, you need hemoglobin to bind the oxygen. CO2, as I said, your plasma loves because it combines with water to make carbonic acid, the bicarbonate buffer system for your blood pH. All right, coming on down. Let's see, let's skip over external because you can read all that as well as I can. I'm trying to make this less painful for us because you can read this stuff as well as I can. Now where I have gas transport, again, big star there. Okay, so Again, how oxygen is transported. The vast majority of oxygen in your blood, please note there, we're looking at, let me get the exact number, 98.5% of your oxygen is carried around in your bloodstream by hemoglobin. That's an important number, big star. The other 1.5% of it's like gas in soda pop. It's dissolved in the liquid. So very little of it is in there. So what is hemoglobin? As I said, you're going to have 4 globular proteins, globins, and each one is going to have a heme particle associated with it. That's iron. Okay. Now each heme can bind 1 oxygen. Well, that means if I have 4 hemes, 4 globins, I got 4 hemes, and that's 4 molecules of oxygen. And as I have there, you have 250,000 of these hemoglobin molecules per erythrocyte. So basically, 1 red blood cell can carry, theoretically, 1 million molecules of oxygen, which is why even if you stop breathing, there's still residual oxygen going around your body. As long as your heart's pumping, you can live still for another 5-6 minutes because your blood hasn't been, unloaded yet of oxygen. All right, so with that, big, huge starters. Okay, I said that hemoglobin has 2 jobs. It's going to carry oxygen and carbon dioxide, but where they bind, 2 different parts of the molecule, one does not interfere with the other. Cannot stress this enough. Carbon monoxide, CO, carbon monoxide is a competitive inhibitor or a competitor for the binding site of oxygen. So therefore, carbon monoxide, if it's present, will compete with oxygen for the same binding site. Problem. Hemoglobin will bind to carbon monoxide 210 times greater than oxygen. So what I'm telling you is that if your body has a choice between carbon monoxide or oxygen, hemoglobin says, screw oxygen, I'm going to carbon monoxide. And if it binds 210 times greater, once bound, it's not going to let it go. That's the whole thing with carbon monoxide poisoning, where you become irritable, then you become sleepy, and then you take the big sleep and you're not waking up again because you basically tied up your hemoglobin and suffocated. All right, moving on down. Let's see. Understand, just so you understand specifics, at 0.1% carbon monoxide in your bloodstream, you already bound 50% of your hemoglobin. At 0.2, you become lethal and anoxic. That's not even 1 full percentage point. Understand that? So carbon monoxide is nothing to play around with. Oxyhemoglobin dissociation curve just means this. Each hemoglobin can hold 4 oxygen molecules. When a hemoglobin is first synthesized, brand new, it takes a lot to get the first oxygen onto a hemoglobin. But once you get the first one on, the second one is easier to get on, the third one's easier still, and the fourth one's a piece of cake to get on. That's what is meant by the oxyhemoglobin dissociation curve. So basically, you want to have your hemoglobin at least have 1 oxygen on it at all times, because that makes it easier to facilitate other ones being added very quickly. All right, carbon dioxide, please note, big star, is transported 2 ways in your bloodstream. The major way, my ginormous star, 90% of it is that carbonic acid system, the bicarbonate buffer system. Okay, that one's huge. The other one is basically going to be where it's bound to either hemoglobin or its dissociated gas, kind of like bubbles going around in the bloodstream. All right, so now, let's see, moving on down, things I have already said. Now, best way to think about hemoglobin. Think of hemoglobin as a flower with 4 petals. Okay, each petal is going to be a globin molecule. As you know, whenever you're in the homeostatic range of pH, you retain your native form. If it becomes out of your pH range, what do the proteins do? They dissociate. Now, when you think dissociate, think like the rosebud opens up to make a rose. And then when you get the right pH, it'll go back to being a bud. If you can think of that as hemoglobin, then the rest of this won't be so hard, okay? But think of it as a bud that can open up to a flower to go back to a bud again. Hemoglobin is designed to go to native form, denature, and go. That's its job, all right? So, now, what I'm going to try to describe to you right now is called the chloride, the chloride bicarbonate exchanger. All right, here we go. You understand that carbon dioxide in water will make carbonic acid, and it dissociates to make hydronium and bicarbonate. That happens naturally. Problem is, it happens very slowly. Whenever you're in your body, a living system, you don't have time for this to go. So as a red blood cell is basically is producing carbon dioxide or carrying carbon dioxide, your blood doesn't want the carbon dioxide in the hemoglobin. It's not accessible. It wants it in the plasma so it can make bicarb, so for your blood pH. Well, how the hell do we get it out? That is the chloride bicarbonate exchanger. Bicarbonate is HCO3 -1. Chloride is an anion, -1. The bicarb is actually, the bicarb and the hydronium is facilitated at a faster rate, created faster because there's enzymes in the red blood cell which actually take the slow pace of adding CO2 to water to make the acid. It speeds it up. It catalyzes it inside the hemoglobin. But as I said, we want the bicarbonate outside. We don't want it inside. Well, HCO3 is a -1. Chloride is a -1. And what do? They switch places. This is called the Bohr effect. It's done through a pump. Okay? Now, by doing that, though, chloride has a negative charge. As chloride is moving in, it's going to attract things of a positive charge to the outside of the red blood cell, namely H+, hydronium. As hydronium builds up, pH drops. When pH drops, proteins denature and the flower opens up. I can pick up oxygen. As I get away to where the pH starts to get back into homeostatic range, what happens? I renature, I carry it around, and I drop it off whenever I denature again. That's more or less in a nutshell how it's going to work, okay? And I don't want to go too crazy technical with it. All right, but that's all called the Bohr effect. Now, down on the bottom of the page where it says venous reserve, big star. As I was saying, oxygen can remain circulating in your blood because, again, your blood is normally somewhere around 98% saturated, 99% saturated with oxygen whenever you're healthy and just breathing normally. So there you have a lot of oxygen attached to your hemoglobin. So therefore, whenever you stop breathing, let's say you were trapped under ice. Let's say that you had an obstruction, you were choking, things like that. Understand, you still have enough oxygen as long as your blood is pumping to continue, 4, 5, 6 minutes still of life. After that, the parts of your body that require oxygen the most start to deteriorate, namely central nervous system, brain, things like that. They start to go really quick. They need to be bathed in oxygen. So if you ever did CPR, let's say you trained for CPR, say 20 years ago, what they used to tell you was you would do 5 pumps of a heart and 2 breaths. 5 pumps of the heart. That's not the new one. If you follow the new CPR method, it's rapid. It's rapid pumping of the chest, and then you blow once or twice, then rapid pump. The point is, you have enough reserve to keep you going. Hopefully, then paramedics or somebody shows up with oxygen then. All right, moving on. Let's continue on. Adjusted rate to your oxygen consumption, as I have there, is due to the ambient pressure of oxygen around you. That's the whole altitude thickness thing that I mentioned. Temperature, as you know, as anything increases temperature, processes occur faster. So as your body temperature increases, you will increase oxygen unloading and loading. That's like when you were on a treadmill working out type of thing. The Bohr effect I already went over. BPG, bisphosphoglycerate, means this. Red blood cells, you understand, do not have a nucleus. They don't have mitochondria, but they still have to have energy. How do they do this? They have to go back to glycolysis. If you remember back when we went over aerobic respiration, the first step is glycolysis, where you took glucose, you broke it down, you made then pyruvate, which then could then be created into other molecules. Right after pyruvate, it becomes BPG, bisphosphoglycerate. So what I'm trying to tell you is, in order for a red blood cell to get energy, it has to do it through glycolysis. Well, this makes sense. Glycolysis is anaerobic. Hemoglobin carries oxygen. Why would you want the molecule that's carrying oxygen to be aerobic, so you'd be using the oxygen you're carrying and run out of it before you got to the destination location in the body to drop it off? That was the analogy I gave you weeks ago where I said, oh, there's a fire over there. Here's a bucket of water to put it on the fire. But before you go, I'm going to punch a hole in the bottom of the bucket. By the time you take the bucket of water over to dump it, there's no water in the bucket. That's what you're trying to avoid, which is why red blood cells don't have the mitochondria to do this. They basically have to work in an anaerobic environment. All right, meaning their jobs. All right, so now the next thing you see is respiratory rhythm and blood gases. All these numbers, understand, are nothing I'm going to test you on, but they're more whenever you go for, say, MCAT type things. You're going to have to know homeostatic ranges. So pressure of oxygen normally in your blood is 95 millimeters of mercury. Carbon dioxide, 40 millimeters of mercury. The main one you need to know, though, for me, big star, pH, you should know should be at 7.4, plus or minus 0.05, meaning between 7.35 and 7.45, you're okay. Anything below 735, you go into acidosis, meaning that your blood has become too acidic, which means nighty-night, you're in a coma. Anything over 745, you go into alkalosis, which means the blood has become too basic, and you'll start going over all over muscle tetany. Basically, your muscles will start to contract uncontrollably, and they won't stop until you come back down within homeostatic range. Now, understand it takes a lot to knock you out of that range because your blood doesn't not only have the bicarbonate buffer system, it has an albumin protein buffer system. It has the heme. I mean, there's 4 different buffer systems for your blood to keep you in range. Hands down, though, the most prolific is the bicarbonate because you're always making carbon dioxide. You can always make bicarb, HCO3-. All right. Let's see. So again, remember that for respiratory rhythm, the most important thing is excessive carbon dioxide. That then makes more carbonic acid, which then in turn drops the pH, which is a secondary factor. Lower pH is what's going to make you increase your breathing rate as well. So again, oxygen doesn't really play a major role here. But as I mentioned, if you have hypoxic drive, anoxic means no oxygen, hypoxic means low level. So somebody has like pneumonia, chronic emphysema, that type of thing, constant labored breathing, then yeah, that would be a hypoxic drive, but that's not the quote norm, for human physiology. For oxygen imbalances, I show you the different things like hypoxemic hypoxia, ischemic hypoxia, anemic hypoxia, histotoxic hypoxia, cyanosis. Again, it's just more vocabulary. Just memorize the vocabulary. The more important one, obviously, would be cyanosis. We talked about that with integumentary skin turns blue due to low level oxygens. Histotoxic hypoxia is due to a metabolic poisoning, usually cyanide poisoning. When somebody is given cyanide, keep in mind the respiratory system is working fine, but cyanide prevents the target tissues, from using oxygen. So basically, they starve of oxygen. That's how that works. Now, that last part you see here is going to be diseases of the respiratory tract. You don't have to memorize them all, but the ones I'm talking about, you have to know. The first one you see there, COPDs. You have to know the COPDs. You have to know the difference between asthma, bronchitis, and emphysema. Again, you can read it, memorize it. It's all spelled out for you, but you have to know it. Coming up now, bronchogenic carcinoma. It's just that's the fancy word to say lung cancer. Again, it's good to know, specifically if you plan on going into pulmonary sciences, pulmonary biology, but for my exam, pneumonia, you should know. It's a common thing. Tuberculosis, I already talked to you about. You should know that one as well, too. RDS is respiratory distress syndrome. That's when you have the lower levels of pulmonary surfactant and the alveoli collapse. That one I talked to you already before. You should know that one. Respiratory failure means you can't get enough oxygen. Again, it's just a common name. Pulmonary embolism means you have a blockage, which means then the pulmonary artery basically pops. Kind of one of those things where you have an intense pain in your chest. You're here 1 second, dead the next. You're like dead before you hit the ground type of thing. Pulmonary edema is just swelling around it. The only last one on the list you should note, star and bedazzle, is cystic fibrosis. Cystic fibrosis occurs because of faulty chloride pumps that are occurring in the epithelium. What's supposed to happen is, think about mucus. Mucus is watery. The reason why is that the chloride pumps, as chloride is being moved across the membrane, water is coming out, therefore making the mucus watery. When somebody has cystic fibrosis, that chloride pump is faulty, so therefore you don't get that exchange. You don't get the water coming out in the mucus, so now the mucus becomes cakey, and therefore it basically becomes almost like paste, and therefore you can fill up the chest. Other problems with faulty chloride pumps with cystic fibrosis can occur in your pancreas and your salivary glands, but people, when they think about cystic fibrosis, they think usually about lungs, but it does affect other tissues as well because of the faulty chloride. So ends respiratory system. My last 2 lectures for you. I know you're sad because I know you're tired of looking at me, but so the last 2 lectures I'm going to go over are going to be then for reproduction, going over male, female, and then the menstrual cycle, all in one big lovely lecture. And then the last thing would then be going over the basics of genetics that you should know for your last test, and the basics of how to do Punnett square and genetic anomalies that you'd be liable for. A lot of them we already talked about before. All right, so I'll see you next time for reproduction.